There, we are going to talk about surface area to volume ratios in cells and osmoregulation. So we're going to take a few look at some of the examples in the body of surface area to volume ratio. So let's take a quick look at that. Let's see. So the first example is with animal nutrition in our digestive system. So when we're looking at our digestive system, that is definitely taking advantage of the surface area to volume ratio. So a very good example of surface area to volume ratio. So the digestive system has many different compartments. So think of it as a tube that runs through your body, and the food that we eat needs to get absorbed across the cells that line our digestive system. So the alimentary canal and the accessory organs all make up our digestive system. And food is moved along through the digestive system through peristalsis, and then we've got sphincters that kind of help to regulate the movement of that material through our digestive system. But we're not going to get into all of those details. We really want to just focus on the surface area to volume ratio examples when we're looking at the digestive system. So food is going to enter your digestive system through your mouth. It goes down your esophagus. It gets into your stomach where it gets broken down. And then it goes into the small intestines where the nutrients are absorbed. They move into the large intestines where the water is reabsorbed in there as well. So a big tube that runs through our body. When we get the food into our stomach, we also have chemicals that help to break down those materials so that we can get to all of those macromolecules that we need and we can get those absorbed into our body. So different things are going to happen in different stages in the digestive system. So when we're looking at our stomach, we've got gastric juices that are made up of both hydrochloric acid and pepsin. But we also kind of need to think, well, why doesn't our stomach get digested by those chemicals that are in our digestive system? So we kind of need to take a look at exactly how that happens. So again, why doesn't gastric juice destroy the cells that make them or damage the stomach? And that's because there are cells that secrete the hydrochloric acid, but they're not secreting it as hydrochloric acid. They are secreting it as hydrogen ions and chlorine ions, which when they get to the stomach, then combine to form hydrochloric acid. And then we also have chief cells that are secreting what will become pepsin, which is a digestive enzyme that breaks down the protein, but it's not released from the cells as pepsin. It's released as another molecule called pepsinogen, which is an inactive version of pepsin. And so when the pepsin is secreted by those cells, it makes its way to the stomach where the hydrochloric acid will activate the pepsinogen, convert it to pepsin, and then it becomes a digestive enzyme. So those are the reasons why they're not destroying the cells that are producing those things, because they're not producing them as those actual molecules. And then the stomach itself, the lining of the stomach is covered by mucus that protects it from those gastric juices. So if we look at the lining of the stomach is very squiggly. So if you look at, can't really see it that well, but if you look up here in the stomach, you've got all of these folds of the tissue that lines the stomach. So those folds, those wrinkles, are actually increasing the surface area of the lining of the stomach, which allows for more secretion of those digestive enzymes, more absorption of some molecules that can be picked up and absorbed in the stomach. And then we also not only have those folds of the epithelial tissue, we also have all of these little holes, basically, that also are increasing the surface area. So we can see all of these cells that line all of these little tunnels that are reaching in to the lining of the stomach, which again is increasing the surface area to volume ratio of the stomach. So we can secrete more digestive enzymes. The stomach can actually stretch because it has all of those folds, and it just makes it more efficient, taking advantage of that surface area to volume ratio. So the stomach itself is an example of how we can increase the surface area of that particular organ to increase its efficiency of how it works. The cells that are also secreting the hydrochloric acid and the pepsinogen are also kind of embedded in these little wells at the bottom, and then they get secreted into the stomach, where they're then going to a hydrochloric acid is going to combine and activate the pepsinogen. So all of that is happening in the stomach. Again, taking advantage of that surface area to volume ratio. Then when we look at the small intestine, so the food will move from the stomach into the small intestines. The small intestines themselves are really, really long. They are about 20 feet long, or 6 meters in length. And that length allows for lots of surface area to absorb all of the nutrients that we need to absorb. So it's really, really long, so that it can, as the food moves through it, it can absorb the nutrients. So that in itself is taking advantage of the surface area to volume ratio. And then this diagram is just kind of showing you that different foods are absorbed in different stages as it moves through the digestive system. So carbohydrates will begin their digestion in the mouth, and then a little bit more breaking down in the stomach, and then they get broken down further and absorbed in the small intestines. The protein doesn't start getting broken down until the stomach, where the pepsin is going to break it down. And then it gets broken down further as it moves through the stomach and the small intestines. When we look at nucleic acids and fats, that digestion primarily takes place in the small intestines. So different food macromolecules will begin their digestion in different stages of the digestive system. Just kind of tying this into our previous unit where we looked at all of those macromolecules. So the duodenum, some people pronounce it duodenum, that is where we've got digestive enzymes that mix with the food that's entering the small intestines. And because the stomach is acidic, we need to kind of neutralize that. So bicarbonate is going to get secreted into the small intestines as well to kind of neutralize it, so that it can the enzymes that are then secreted can actually work as well, because they're going to be more efficient at that particular pH. And we haven't gotten into enzymes and enzyme activity yet, but we will see that in a later unit. So we're not, again, going to get too much into all of the things that get secreted into the small intestines, but enzymes will get added into the food that is in the small intestines so that it can be absorbed. But again, the small intestines are really, really long, so that we can take the time as it moves through to pick up those nutrients. But also, the small intestines have microvilli and villi in the walls of the small intestines, again, increasing that surface area to volume ratio. So if we look at the lining of the small intestines here, we can see it's all squiggly, kind of like the stomach, but actually even more so. And those squiggles, the wrinkles, are called villi. And again, those wrinkles are taking advantage of the surface area to volume ratio. So it's increasing the surface area of the lining of the small intestines. And then if we look at a cellular level, those microvilli, or the villi, are lined with cells that have microvilli, and the cells themselves have these little bumps on them. And those little bumps can then absorb more nutrients as well as they move across the lining of the small intestines. So the villi increase the surface area of the lining of the small intestines. The microvilli, even more so, they're increasing the surface area at a cellular level so that they can get more nutrients into the cell from the lining of the small intestines. And when we look at moving things across the membrane, Some things move in with facilitated diffusion, some things move in with active transport. So different molecules are going to get into the cells that are lining the small intestines, either through facilitated diffusion, simple diffusion, or active transport, depending upon the concentration, things like that. So fructose is going to move through facilitated diffusion down its concentration gradient. And other nutrients like amino acids, small peptides, vitamins, most glucose, those are going to be pumped against the concentration gradient using active transport. So again, active transport, when you're going from low concentration to high concentration, that's going to require energy in the form of active transport. So again, different molecules are going to move across the membrane, depending upon their concentration, how they are going to get into those cells. We're not going to really get into the liver and all of that, the role of the liver in that. So moving on to fat absorption, the fat molecules are really big. And so they actually need to get broken down in order to get into the cells that are lining the digestive system, the small intestines. So they get broken down and then they get brought into the cells. They will reassemble where they're then encased in these little clusters that can then go into our bloodstream and circulate throughout our body. But again, the lining of the small intestines has those microvilli that help to get those molecules into the cells and then into the bloodstream and then to wherever they need to go. The large intestines is going to then continue and finish up the digestive system. The primary function of the large intestines is to reabsorb the water. But again, that's kind of little bumps in there to increase the surface area of the large intestines. The large intestines is primarily storing the waste, but reabsorbing the water in our digestive system. Okay. So it's recovering of the water that's entered the alimentary canal. Okay. So that digestive system, a good example of surface area to volume ratio, but then we're also looking at molecules moving across the membrane into the cells that are lining the digestive system. So we're kind of looking a little bit at active transport and facilitated diffusion there. Another example of surface area to volume ratio is looking at circulation and gas exchange. So the circulatory system, we can see the surface area to volume ratio as well. So with the circulatory system, we're looking at the diffusion of oxygen and carbon dioxide across the cell membrane. So when we look at how the blood flows through the body, we're kind of looking at our arteries and our veins, and then those arteries and veins are going to branch. So when the blood leaves the heart, it leaves the heart through the arteries, and then those branch into arterioles, those then branch into capillaries, and then the capillary beds are where we've got chemical exchange between the blood and those cells where it's carrying the nutrients. So we're going to have oxygen exchange, we're going to have nutrient exchange, we're going to have carbon dioxide exchange, we're going to have waste exchange at the capillary beds. And then those capillary beds will combine back together into venules and then veins, and then they return to the heart. And so that's kind of how the blood circulates through our body. And so all of those things are exchanging in the capillaries. So that will occur through both blood pressure and osmotic pressure will help with the movement of molecules across the membrane. And so again, we're going to see a lot of diffusion because the carbon dioxide is going to diffuse into the blood where it's in low concentrations from where it is in high, and the oxygen in the blood is going to diffuse out to where it is in low concentration. So high concentration to low concentration. So the carbon dioxide and the oxygen are typically diffusing through simple diffusion, but there's blood pressure involved in that exchange as well. But we're not going to get into all of those details. We're going to focus primarily on the diffusion of those molecules across the cell membranes. And then there are respiratory surfaces that will help with that gas exchange. And so when we're looking at our respiratory system in conjunction with our circulatory system, again, we're looking at diffusion across the surfaces of the respiratory system. So if we look at aquatic animals, they require oxygen, obviously, and they're going to get that through their gills. And they rely on the water because we've got the dissolved oxygen in the water. They're relying on the water flowing over the gills and then the gas exchange to take place in the gills. I'm not going to get too much into that counter current circulation, but again, they've got those capillary beds that are going to be lining the gills so that we can get gas exchange. We're going to focus more on the lungs, which is very similar to the gills, again, where we've got that circulation, that the gas exchange across the lining of our respiratory system. So like the gills, we have lungs. That is where we're going to have our gas exchange take place. And again, if we look at the lungs, we're seeing a large surface area to volume ratio. So that's going to take into consideration as well. So at the lungs, when we breathe in air, it goes into our lungs and it gets branched. And then at the very end of all of those branches, we have alveoli. And surrounding alveoli, and they kind of look like these little broccoli structures. That's where the gas is going to fill in there. And surrounding that, we've got capillaries. And so the capillaries increase the surface area and the alveoli increase the surface area. So now we've got lots of surface area to exchange the nutrients, the gases that we need and we need to get rid of. So we need oxygen to go into our lungs and then into our capillaries. And we need the carbon dioxide to go out of our capillaries into the alveoli so we can breathe it out. So the gas exchange is going to take place in those alveoli along those capillary beds that are surrounding the alveoli. And so we've got lots and lots of branching going on. So we've got lots and lots of surface area to be efficient at that gas exchange that's necessary. So our lungs are another example of taking advantage of that surface area to volume ratio of those cells. So the circulatory system, again, those veins are going to branch and branch and branch until, or I should say arteries are going to branch and branch and branch until we get to the capillaries. And then those capillary beds are branching so much that they are increasing that surface area. And then they come back together and then go back to the heart. So the circulatory system also takes advantage of the increased surface area to volume ratio to be more efficient at getting those gases exchanged. And the last thing that we're going to take a look at is we're going to take a look at osmoregulation and excretion. We're just going to do that super quickly. Looking at that, osmoregulation is when we're looking at maintaining solute concentrations and keeping those in balance. So organisms need to be able to maintain a certain amount of water in their body. And so osmoregulation is really just that ability to maintain the amount of water that we need to keeping that uptake of water in balance. Osmolarity is the solute concentration of a solution determines the movement of the water across a selectively permeable membrane. So we've been talking about molarity, we've been talking about solute potential. Osmolarity is just looking at that solute potential as well. So it's just another way of looking at osmosis and the movement of water and the ability to influence the movement of that water. So it's just another vocabulary term to be familiar with. But think of osmolarity as simply the molarity and how the molarity of that concentration of water will cause the water to move across a selectively permeable membrane. So we're just simply looking at the movement of the water. And then there are different organisms that will be using different techniques to maintain that osmoregulation. So osmoconformers are just going to be those organisms, marine animals, that are going to be isoosmotic with their surroundings, which means they're going to be in an environment that they are isotonic with, so that they don't need to worry so much about the water movement across their membrane. And then osmoregulators are going to be ones that can use energy to maintain that balance. So they can either take in more water or get rid of that excess water, so they can actually regulate their water levels. When we look at nitrogenous waste, which is basically our pee, that's going to kind of reflect the physiology of the organism and its habitat. And we're not going to get into that too much, but our proteins, when we break down proteins, that's going to give off nitrogenous waste, and we need to get rid of that nitrogenous waste, which is primarily what we're going to excrete as urine. So some animals are going to excrete that ammonia in different ways, and they're going to have more or less toxicity depending upon how they are excreting that waste. So different animals are going to be excreting that extra nitrogen in different ways. And so there are different forms of nitrogenous waste depending upon the different animals. So some animals will excrete ammonia, some urea, some uric acid, and they're going to have different toxicities, and they're going to require different energy to excrete those different wastes. But we're not going to get too much into that. Just know that we are excreting that extra waste. And when we as humans excrete that extra waste, that we're going to do in urine. And our kidneys are another example of increasing the surface area to volume ratio, and also looking at that osmoregulation and getting rid of those wastes that are building up in our body. So our kidneys will take advantage of nephrons that are inside the kidneys, and they will run alongside of our capillaries, and they're going to filter the blood and get rid of and excrete the waste that's built up in there. And that's going to take advantage of, again, surface area to volume ratio and diffusion and osmosis across the cell membranes. But again, we're not going to get into that too much, but looking at the nephron, it has different areas that's going to absorb different materials. So things will diffuse into and out of it in different locations to get rid of the waste, but reabsorb the water, keep the good things in, and get rid of the bad things. And our kidneys will work on doing that. So it's a step-by-step process for filtering the blood. Different parts of the kidney and the nephron are going to absorb different molecules and get rid of different molecules and excrete those in our urine. But we're not going to get into that too much detail. We will leave that for an anatomy class. But the water will diffuse out, and then some of it gets reabsorbed as it does so. And so it uses diffusion and osmosis to get rid of the waste in the kidneys. So those are all examples of surface area to volume ratio and osmosis and diffusion, and applications into how they are applied to scenarios within our body. I hope that was helpful, and I will catch you guys next time.